Welcome, colleagues. Let's think about a concept you probably thought was settled years ago. For decades, dietary fiber was basically dismissed, right? We thought of it as just simple, inert roughage, just waste passing straight through the human body. Well, today, whether you're an analytical chemist, a clinical gastroenterologist, a food technologist, or a toxicologist, we are going to completely reframe that idea. In this explainer, we're going to look at the precise molecular mechanics of dietary fiber. It is actually a highly bioactive system. We'll explore how it alters cellular signaling, impacts systemic disease, and why it demands very rigorous scientific dosing.
Here is the translational science arc we'll be taking today. We will quickly cover Section 1, Beyond Roughage, the Fiber Paradigm. Section 2, Structural Chemistry and Classification. Section 3, Gut Fermentation and SCFA Signaling. Section 4, Clinical and Metabolic Health Effects. Section 5, The Sucrase Deficiency Paradox. And finally, Section 6, Dosing, Toxicology, and Upper Limits.
Okay, let's start with Section 1, Beyond Roughage, the Fiber Paradigm.
I want to explicitly address the analytical chemists and food technologists tuning in, because how we define our subject matter changes everything. Historically, early nutritional science was profoundly misled by something called the crude fiber method. This was strictly a chemist's concept. Imagine treating food with boiling sulfuric acid, strong alkali, water, and alcohol. Obviously, this highly aggressive extraction dissolved away many components, which gave us misleadingly low fiber levels. It really had very little to do with actual human nutrition. The revolutionary shift happened when we finally adopted a physiological concept, dietary fiber, officially recognized by the US FDA back in 1984. Dietary fiber is defined by human biology, not boiling acid. It includes the plant constituents that actively resist digestion by human gastrointestinal secretions. It's exactly what survives the journey.
Right, moving on to Section 2, Structural Chemistry and Classification.
Let's explore the structural biochemistry here. Why are these molecules so highly resistant to our endogenous digestive enzymes? Well, for the biochemists listening, you'll love this because structure is essentially destiny. Look at cellulose. It consists of linear beta-1,4-glucose polymers. Human alpha amylase simply cannot break a beta-1,4 bond. Because we lack cellulase, it remains completely intact. It's water-insoluble, and its main clinical action is decreasing intestinal transit time. Pectins, on the other hand, are polymers of galacturonic acid. They are water-soluble, they form these viscous gels, and they actively lower plasma cholesterol. So the physical property directly dictates the biological fate.
Now, notice we have to include resistant starch here. This is a massive contributor to the dietary fiber pool. Did you know, for our food technologists out there, that as much as 35% of legume starch escapes digestion entirely? It's true. It travels straight to the large intestine and acts as a very potent prebiotic, feeding beneficial colonic bacteria. It is a highly functional subcategory, and you'll find it in everyday foods like cooled pasta and barely ripe bananas.
Let's jump into Section 3, Gut Fermentation and SCFA Signaling.
So, the crucial point is tracing what happens biologically when these digestion-resistant molecules finally reach the lower GI tract. Clinical physicians, let's track this metabolic journey step by step. Step 1 is gastric survival. The hydrochloric acid in the stomach breaks plant cellular structures to release nutrients, sure, but the fiber itself remains untouched. Step 2 is small intestine transit. Interestingly, in vitro studies show viscous fibers can actually inhibit pancreatic digestive enzymes here. Step 3 happens in the colon. This is where the gut microbiome steps up as the hero. Bacteria initiate the fermentation of hemicelluloses, pectins, and gums. And that leads to Step 4, short-chain fatty acid, or SCFA, production. The microbes generate acetate, propionate, and butyrate. This is the exact moment where so-called inert roughage becomes a powerful metabolic signaling system.
And this actually brings up a fascinating ongoing debate for our dietitians and food technologists: energy labeling. Because absorbed SCFAs provide usable energy to the body, fiber is not actually 0 calories. In the US, soluble fiber must be counted as 4 calories per gram on nutrition labels, while insoluble fiber may be treated as 0 calories. This creates some real regulatory inconsistencies, simply because fermentation and energy absorption vary so radically depending on the specific structural chemistry of the fiber.
Which brings us nicely to Section 4, Clinical and Metabolic Health Effects. Now, let's look at the systemic clinical outcomes. How do these molecular interactions actually manifest as disease prevention in your patients? For the pharmacologists and cardiologists in the audience, I have to emphasize this profound hypocholesterolemic effect.
We consistently see a 10 to 15% drop in low-density lipoprotein, or LDL cholesterol, when using soluble fibers like guar and pectin. Why does this happen? Because these viscous fibers, along with lignins, literally bind to bile salts in the intestine, stopping their reabsorption. Since bile acid excretion is the main way we eliminate endogenously synthesized cholesterol, the liver is suddenly forced to pull cholesterol directly from the blood to make new bile acids. It's a brilliant natural mechanism to lower plasma cholesterol.
And the systemic benefits don't stop there. Feeding experiments show that stool weight increases by 3 to 9 grams for every single gram of cereal fiber you add. Fermentation also decreases colonic pH, which limits the uptake of toxic ammonia by epithelial cells. We also see that a high-fiber diet increases peripheral sensitivity to insulin, making it an absolutely crucial non-pharmacological tool for managing diabetes. Furthermore, soluble fiber delays gastric emptying by forming a thick, viscous gel matrix. This promotes a strong feeling of satiety, which is highly relevant for combating obesity.
Public health scientists take note of this powerful epidemiological finding. Correlation and case control studies suggest there's a 31% potential reduction in colorectal cancer risk if dietary fiber from food sources is increased by an average of just 13 grams per day. To put that into perspective, the proposed mechanics are quite elegant. Fiber increases fecal bulk, diluting potential carcinogens. It shortens transit time, which decreases the amount of time the mucosa is exposed to toxic materials, and of course, those generated short-chain fatty acids may provide direct anti-neoplastic effects on the colonic epithelium itself.
Let's transition to Section 5, the sucrase deficiency paradox.
I want to share an incredible clinical mystery documented in Johannesburg, South Africa. It beautifully reinforces everything we've just covered about gut fermentation. Here is the epidemiological paradox. We generally know that rural, high-fiber diets protect against non-infectious GI diseases, like inflammatory bowel disease and colon cancer. But in a specific section of Johannesburg, where both Black and Caucasian populations followed a low-fiber Western lifestyle, the Black population maintained a persistently rare incidence of these diseases. They lost the dietary fiber intake, but they somehow kept the clinical protection. How is that possible?
Well, the biochemical answer was hidden in duodenal biopsies. These biopsies revealed significant differences in disaccharides activity. The Black patients primarily had very low lactase, but also significantly lower sucrase, a mean of 22.8 units compared to 43.5 units in the white patients. Because of this specific deficiency, unabsorbed sucrose was passing straight into the colon. And what happens then? It mimics the exact fermentation behavior of dietary fiber. It generates those same protective short-chain fatty acids we talked about earlier. It is just a fantastic demonstration of functional metabolic equivalence in the human gut.
Finally, let's cover Section 6, dosing, toxicology, and upper limits. Toxicologists and clinical dietitians, this is for you. We absolutely must examine upper dosing limits. Because dietary fiber is a potent bioactive compound, it has a strict physiological and toxicological window. Look at this stark contrast. The recommended intake for adults is 20 to 35 grams per day. Sadly, the average U.S. intake is alarmingly low, hovering around 12 to 15 grams daily. However, there is a very strict upper threshold. Exceeding 40 to 50 grams per day is the danger zone. That is where therapeutic benefits cross over into toxicity, and significant adverse physiological effects begin.
So, what actually happens when we overprescribe fiber? The risks of overconsumption are actually quite severe. We can see intestinal obstruction and dangerous fluid imbalances. Biochemically speaking, fiber-associated substances like phytates will bind divalent cations. This induces severe deficiencies in essential minerals like zinc, iron, and calcium. Saponins can even alter cell membrane characteristics. All of this creates a deeply negative mineral balance, which is incredibly dangerous for growing children, adolescents, and pregnant women. Plus, fiber-rich foods can contain detrimental protease inhibitors that harm populations surviving on marginal diets. This is exactly why precise, targeted dosing is so absolutely critical.
Before we wrap up, I want to remind our multidisciplinary audience to always maintain the highest scientific rigor. As the literature states directly, the possible relationship between dietary fiber and colon cancer remains a hypothesis that must be put to a rigorous test. Epidemiological studies can be misleading. We have to remember that landmark clinical trials, like the polyp prevention trial, found no effect of a low-fat, high-fiber diet on the recurrence of adenomas of the large bowel even after 8 solid years of follow-up. We simply cannot rely on epidemiological correlations alone. We need controlled, precise clinical data.
We've just traced dietary fiber from its basic linear glucose polymers right through its complex role in systemic disease prevention, all the way to its strict toxicological limits. So I want to leave you with this question. How will you leverage the precise molecular mechanics of dietary fiber in your next clinical trial, food formulation, or toxicological assessment? Thank you for joining this explainer. Until next time, keep looking deeper into the science.